my name is Rachel Newsome, and I am a co-founder of BB Therapeutics. And the goal of our company really is to extend and improve the lives of cancer patients. We are a pre-seed stage startup out of here in the University of Florida. So here's a little bit of background on our team. So I am currently a postdoc in the lab of Dr. Christian Jobin. He is my co-founder and co-inventor. And my PhD thesis work, and now currently my postdoc work, is actually what led to our exciting discovery. So Dr. Jobin is a worldwide expert, cancer and microbiome. He's received over $25 million in federal and private funding. He is on the advisory board of numerous scientific companies, and he's the section editor of many journals. Our board chair and advisor is Todd Kruger. He's currently the CEO of another microbiome company. He has deep expertise in early stage drug development. So together, we really have our finger on the pulse of the field in terms of both research and commercial development. So we focus on immunotherapy, which has really revolutionized cancer care and life expectancy in some cancer types in some patients. Just last year, as illustrated on the left in this waterfall plot, 100% of colorectal cancer patients with a specific subtype had complete pathologic response to immunotherapy, with 79% having complete eradication of this cancer. Now, this is called a waterfall plot. I would argue this is more of a lake. Now, high profile work from our lab has found that gut metabolites from bacteria can actually influence the response to immune checkpoint inhibition, which is one type of immunotherapy. And it can do this in distant and immunosuppressive non-responsive cancers. And for us, this is really a key moment in thinking about how we could maybe boost the immunotherapy response. So with the incredible data I just showed you, you may be asking yourself, why do I still have a job? Well, these drugs may represent a $40 billion market, but they actually fail in the majority of patients in cancer types. This is exemplified by non-small cell lung cancer, the leading cause of death by cancer. But only 20% of patients respond to immunotherapy that have this cancer. So a drug that could be used in combination with these drugs to increase response would be an absolute blockbuster. So the main reason for failure, especially for immune checkpoint inhibitors, is that immune checkpoint inhibitors only work if an immune response has been activated. So the drug works by disrupting this PD-1, PD-L1 interaction as illustrated here. And the drug can still work, but not result in any meaningful benefit if the killer T cell, here illustrated by the CD8 killer T cell, is not activated against the tumor. So our lab has isolated a novel small molecule from immunostimulatory bacteria. We call this small molecule BB01. And this provides the initial activation for killer T cells to actually mount an attack against the tumor cell. So our journey to this discovery actually started with a collaboration with Moffitt Cancer Center. So we enrolled 65 late-stage metastatic non-small cell lung cancer patients, and we stratified them by response. And what we found, interestingly, was that there are 6 immunostimulatory bacteria that are actually enriched in these responder patients. And these bacteria are able to stimulate cytotoxic T cells to kill cancer cells. Now, what we further did, and this is really the novel work done in our lab, is that we performed mass spec analysis of these bacteria. And we found one novel small molecule, BB01, which has these immunostimulatory properties. And this small molecule actually directly activates killer T cells. So we have validated the therapeutic benefit of BB01 in preclinical models, and now we are generating next-gen derivatives. So these have optimized activity with better intellectual property rights. So just as a proof of concept, we found that given systemically, BB01 reduces tumor growth by 50% in distant non-responsive lung cancer when given with immune checkpoint inhibitors. And it does this by driving migration and infiltration of these activated immune stimulatory CD8 T cells. And this causes a potent anti-tumor response that would not happen with just PD-1 alone, as illustrated here. Now, we are currently in the process of engineering BB01, and this is so we can enhance the solubility, bioactivity, and also have strong IP protection. We call these next generation derivatives BB0X. And so as a proof of concept, we actually encapsulated BBO1, which is the natural small molecule, in a lipid nanoparticle formulation. And so this improves solubility for intravenous administration. And just 24 hours after administration of these loaded nanoparticles with BBO1 into mice, we saw a doubling of the activated cytotoxic T cells in the blood of these mice. So next generation BBOX has many desirable features. It will be more soluble, more potent, enhanced stability. The advantage of a small molecule drug is, of course, easily defined PK/PD and mechanism of action with a clear pathway to FDA approval. We have solid intellectual property with a strong UF pipeline. We have already filed on these next generation derivatives, and we are currently building on that filing. So, given the large market, there are many competing drugs in this space. However, we believe that BBO1 offers several competitive advantages. So, competing drugs like recombinant interferon gamma and bispecific antibodies, they actually enhance the immune response on many cell types. And this includes mucosal epithelial cells, which are all across the body. And sometimes this is what leads to that severe treatment ending colitis and pneumonitis that you see in these patients. So many of these drugs try to circumvent this toxicity by using direct intratumoral injection. Now, this is highly undesirable for solid tumors and also especially in lung tumors. Now, what BBOX does and BBO1 is they actually target T cells, cytotoxic T cells specifically. So this leads to less immune-related adverse events than our competitors. It allows for systemic administration, and this can be intravenously or even orally, given the stability of our small molecule. Now, recombinant interferon gamma has already been proven to be ineffective in the clinic with severe adverse events, but BBOX offers a different mechanism of action. We're actually not binding to the interferon gamma receptor. We have a different, very novel mechanism of action that is very specific to CD8 T cells, and this offers that activation that is highly specific so we can do systemic administration. So lastly, BBOX we validated in large and immunosuppressive tumors. Cytokine pathway agonists have very poor efficacy in these tumor types, and this includes non-small cell lung cancers, and that's why they're ineffective. So we believe that BBO1 and BBOX will offer advantages in all of these categories. Now, we are currently in the lead discovery phase. So we have ongoing support from the University of Florida, and this will get us to our in vivo proof of concept data on our lead BBOX derivatives. We also intend to submit an STTR grant in the winter to support our startup efforts. Now, what we are seeking is an additional $100,000. This is to further de-risk our derivatives. We want to do benchmark comparison therapy and additional indication testing. This data is crucial and will allow us to seek seed funding to support our commercialization. So in conclusion, although immune checkpoint inhibitors are a $40 billion market, an incredible treatment for some patients, the low response rates really limit the benefit for most patients. Our novel small molecule drug presents the opportunity to double these response rates, favorable delivery, less toxicity, and ease of manufacturing than competitors in this space. We have strong intellectual property, supported by UF Innovate, as well as a team of leading experts from the University of Florida. We are asking for a $100,000 investment to further de-risk these leads and prepare for seed funding. And I'd be happy to take any questions.